I'm gonna pull up chat so that if you guys start chatting, I can read what you say. All right, so for chapter 1, we discussed what microbes are. We talked about what some of the acellular microbes are. So the ones that are acellular, they do not qualify as having cells or being cells. So remember, a cell is basically going to be a membrane bound, basically say a discrete unit of life or whatever, and it can reproduce a copy of itself. It has everything that it needs to make a copy of itself. So acellular microbes, they don't qualify within this. Okay, so these would be our viruses and our prions. We'll get into more detail on those guys in chapter 7. And then we talked about the eukaryotes. So eukaryote, A being without and karyote being a nucleus. So these guys do not have a nucleus. That's like their defining feature. They don't have any organelles at all, but we'll talk about that a little bit more when we get into chapters 4 and 5. So, but very literally, the word eukaryote means no nucleus. They're likely the first organisms on the planet, and then organisms that had a nucleus. We're going to talk actually a little bit, we're going to touch a little bit on how those guys came to be and how we believe that they came to be. There's a ton of supporting evidence on the theories on how eukaryotes or true nucleus organisms came to be. So there's that. Microbes are probably the largest contributors to the atmosphere, and definitely in the beginning of time, they created oxygenated atmosphere. And now when we talk about oxygenated atmosphere, excuse me. Now, when we talk about oxygen and atmosphere, of course, plants play a very big part in maintaining that, but microbes are still our heavy hitters for that. So microbes largely responsible for our oxygenated atmosphere. Pathogen is just any microbe that is responsible for causing illness. Right. And then we talked a little bit about Pasteur and his ability to disprove spontaneous generation. So remember, spontaneous generation, I'm not going to write it out on here, but just to remind you guys, spontaneous generation was that theory that like life could just come out of wherever. And if you had like a steak that you left out on a countertop, the reason that it is smelly and has a nasty, gross film on it and has mold on it and gets maggots on it is because that's just naturally just going to happen on its own. But that's not true. We have life that begets life. So there were microscopic organisms now that are multiplying, for example. And that, his concept that he named that's called germ theory. That germs were there and that they're things that he couldn't see, and he called them germs, and that's what germ theory is. And those are our microbes, they're the germs. We also introduced this concept of aseptic technique, which we're going to get into a little bit more detail about what aseptic technique is when we get to using aseptic technique in the laboratory. But aseptic technique was very first employed. Why was that so thick? And, by Joseph Lister. So he started using carbolic acid or phenol in his surgical operating theaters in order to sanitize those operating theaters, so he would decrease disease and contamination of wounds and things like that during his surgeries, and that was hugely beneficial to preventing infection post-surgery. So he was the first one to utilize aseptic technique. And what aseptic technique really is, our basic concept, what we need to get out of aseptic technique is really just preventing contamination. That's anything that you're doing to prevent contamination. So when we work in the lab, we want to prevent contaminating our environment with our microbes that we're working with, but we also want to prevent contaminating our cultures with things from our environment. It goes both ways. So preventing contamination is the take-home message with aseptic technique. Then we introduced taxonomy. And domain, and then kingdom, phylum, class, order, family, genus, species. And one way you could remember that would be King Philip came over for good spaghetti or something like that, whatever works for you in your mind. But those were like the original ones back whenever I was like in grade school. And then domain was tacked on to the front of it. So yeah, or so you could ask, there's a question, did King Philip come over for good spaghetti? You could think of it that way. So tack that domain on the very front. We have to actually know the 3 domains. Why is it when I draw a line, it's so thick? This one isn't thick. I don't understand. Whatever. It doesn't matter. Anyways, I don't feel like I felt like I felt pushed pretty hard on that line too, but the other ones I didn't. Our 3 domains, we would say would be bacteria, archaea, and our third one would be eukarya. So those are our 3 domains. And then we have binomial nomenclature. And what that is, this is going to basically be the fact that we have the 2 words that make up a name of a species. All right, we're always going to capitalize the first one. The second one starts with a lowercase letter. If you're typing it, you'll italicize it. If you are writing it by hand, then you will underline it. And this is where we get those names like E. coli, which you would technically underline if you were writing, or Staphylococcus aureus. And you would underline it if you were writing it by hand. If you're typing it again, be sure you would italicize it. Those are the rules for binomial nomenclature. So we'll move on next. That's the wrong thing. To chapter 3. In chapter 3, this is basically getting us into the idea of the tools of what we're going to use to study microbiology. We start off with the 5 eyes. So our 5 eyes. 5 eyes. We have these kind of are going to form our basis for the things that we just generally talked about in chapter 3. But the first step in the 5 eyes is inoculation, then incubation, then we're going to do isolation. I don't know why. Isolation. There we go. We're going to do inspection and then identification. Okay. So those are 5 eyes, and they are kind of stepwise. They happen in an order. Inoculation is putting whatever your sample is, introducing your sample to a sterile media. So we're going to actually start culturing it. So this is basically the culturing step. Incubation, we're putting it at the favorable temperature to grow it. Isolation kind of speaks for itself. We're going to separate out the mixture into the individual parts. So what that means is if you have multiple microbes in there, we're going to separate out your sample enough that you can get the individual microbes separated from one another. So this is literally just separation step. Inspection, we're going to be doing tests to try to figure out, like, does it grow on this media? Does it grow on that media? When I put it through the staining process for gram stain, what's our result? When I put it through the staining process for the endospore stain, what's the result? Those sorts of things. These are our tests. And then identification is like using the results to figure out what it is. Then literally everything that we learned about after that did apply pretty directly to that. 2 things that we learned about while we were talking about the 5 eyes and how they're going to apply to microbiology were a couple of things. Were differential and selective media. And yes, I do believe if you can at least remember what the words mean, that they should tell you what they are and what they do. Differential media, it just shows you the difference between organisms. And so, yeah, it shows the difference. Usually, this is because we'll get a color change in the media. So you either turn the media a color or you don't, or you could turn it any number of colors. And those colors aren't the end all be all result. It's not like you grow it on this media and it turned red and red is the result. No, you need to know what that color would mean. So, what would the color mean? They're always a sign of a metabolic biochemical difference. So it's usually based on a byproduct in a metabolic pathway. So shows difference, and you could say it shows the difference based on biochemical byproducts. That's what you would say that it literally does. And that's shown usually by a color change in the media. Selective media, on the other hand, this one is selects um, what grows and what doesn't. So it's basically what selective media is going to be, right? And so there's something in it that will prohibit the growth of some organisms, therefore allowing the growth of others. Or it might have very selective ingredients for just certain groups of bacteria. But whatever it is, only some things can grow on it and other things can't. That's not the case with differential media by itself. If a media is just differential, anything could technically grow on it, but it might have a color change involved. Could you have media that is both? Of course. If it has something in it that prohibits the growth of another organism, it is selective. And if it has something in it that would cause a color change in the media because it makes a certain byproduct, it's differential. And you can have both of those things going on in one type of a media. Media is just the stuff that we're growing our bacteria on. That's what the term means. Okay. All right, so here's our next thing that we will talk about: enriched media, fastidious organisms, and growth factors. Enriched media is media that has a growth factor added to it. A growth factor is something that that species cannot make for itself. It has to have it added to its diet. So things like we have with vitamins, we can't make that for ourselves. We have to have it in our diet. So a growth factor is something that the organism needs in its diet that it can't make for itself. So it can't make it, we have to add it. And if an organism is that way, if it is the type of organism that is going to need a growth factor added to its media, then we're going to call it fastidious. So it's a fastidious organism that requires a growth factor be added to its media. That media has a growth factor in it to help those fastidious organisms grow. We call that media enriched. All right. Next, we talked a little bit about microscopy and how microscopes work. We talked about the flow of light in a microscope. You're basically going to have your light bulbs, and that's going to make light come out. And then that's going to travel up through the actual specimen on the slide. Here's your nice little slide. And then you will have a lens that is looking at that first. And this is the objective lens. And then that'll travel up through the part that you'll actually be looking through, which is your eyepiece. Okay, so that has another lens in it too, the ocular lens. I know that's really fancy and it looks very beautiful, but you get the idea of how the light is traveling through the actual microscope. Okay, so that's how it goes. That real image is the one that sees that the specimen is made from the specimen first. So just think of it that way. So if we have the pathway of light, please go away. Please go away. If we have the light going to the specimen, going to the objective lens, then to the ocular lens. Right, so that's the steps of the light. This step here, this part is the where we create the real image. Okay. This part here, because we're going through another lens, all right, before you see it in your ocular eye, in your eyepiece. This part here, we're creating that virtual. And one way, like I've said before in class, to help you remember that the virtual image is what's going through the ocular lens, ocular eye. We kind of know what that means. Virtual, it looks a lot like the word visual. So that can help you remember that the virtual image is being made at that visual step where you're going to be looking at it. Okay. That'll help you remember that. Oil immersion. This just reduces light scatter. When we're reducing light scatter, another word for light scatter is refraction. Okay. Heat fixing is just a way that we would be using heat to literally fix. I mean, it says it in the name, right? Heat will literally fix your sample to your slide. It'll also kill that organism. So those are two, probably the two big things that we would have that we need to know about heat fixing, which would be, one, it's going to fix it to the slide, fix it or adhere it to the slide. And two, kill the organism so that you're not dealing with something that could be contaminating your environment or you or whatever. If you're looking at infectious organisms, that's going to be a useful thing. It also, like, other than just fixing it to the slide, it also fixes whatever parts are in that organism into place, like permanently. So the parts in the organism also aren't moving around or whatever. So there's that another part, but these are like the two big take home messages with why we heat fix things. Okay. So there's that. Staining. Staining, the probably the biggest use for staining is to provide contrast so we can see our specimens a little more clearly. There's two types of stains, negative and positive stains. Typically, when we're talking about these, are positive. These are typically our basic stains. Our negative, typically acidic. Okay. So the fact that your cells, which we learned about in chapter 2, with the fact that your cells are covered with this lipid bilayer that consists of, your phospholipids. And on the outside of your lipid bilayer, you've got the phosphate heads. And then on the inside, you have the hydrocarbon tails, which are hydrophobic, right? This is the phospholipid bilayer that makes up cell membranes. All right. So if this is on the outside, and we know that phosphate is very negatively charged, opposites attract. So your positive or basic stains, these guys are going to be attracted to that. This definitely says attract, okay? So because of that, our basic or our positive stains usually stain the cells themselves, whereas our negative stains are repelled by the negative surface of the cells, and they will stain the background. All right, so that's how that's going to work. And then we mentioned a little bit about differential stains, and we just introduced our differential stains. We didn't talk too much about how they work or anything like that. We will definitely be getting into the details on that in chapter 4. But at least for now, be aware that these are differential stains. They're not simple stains. A simple stain uses just one literal dye, just one dye, and that just stains the whole cell. And it doesn't really tell you if it has a structure or not. These stains here, the gram stain, the acid fast stain, the endospore stain, the capsule stain, are some stains that we'll be doing in the laboratory that will tell you whether your organism is positive or negative on the gram stain, whether it is acid fast positive or acid fast negative, whether it has endospores or doesn't have endospores, whether it has a capsule or it doesn't have a capsule. And those stains will literally tell you the difference between that just by doing the stains and looking at them in a microscope. And so that's how differential stains will work. So moving on to chapter 2. In chapter 2, we basically gave our chemistry review. Big take home topics for that. We talk about the different types of bonds, right? So we talked about covalent bonds. These were sharing electrons between two atoms. Ionic bonds, where we are transferring or donating an electrons completely, permanently. And then hydrogen bonds, which there is no electron movement in the actual hydrogen bonds themselves. These are entirely attraction-based. Okay, so, and always a hydrogen bond. So here's one big point. They don't share, move electrons. Another big point, it's attraction-based. The other final take-home message, always one of the atoms involved in the attraction is going to be hydrogen. It's in the name. All right, so there's that. Then we talked about organic molecules. An organic molecule is just a molecule that is carbon-based, and it must have hydrogen associated with carbon in some degree. All right, we also talked about polarity and what that is and how that works. And then we talked about like charging of the molecules and how that's going to affect how it behaves with like water and things like that. But polarity essentially, it's going to result from our electronegativity of certain atoms, and very electronegative atoms. Why? What's happening? Oh, it's switched to eraser. All right. Electronegativity. So our very electronegative atoms will hog electrons, right? And our best example of that is oxygen. It's really notorious for it. All right. And it often hogs from hydrogen, which will get a negative charge, or will get a positive charge as a result of that. So oxygen will get a negative charge. We'll say polarity. That's probably the more chemically correct terminology to be using at this point. Polarity. All right. So if the hydrogen is attached to the oxygen will hog the electron from the hydrogen. The oxygen then gets a negative polarity. The hydrogen gets a positive polarity. All right, so that's how that usually works. That's water molecules, and we'll see that literally happening within water molecules. Why? We'll get that polarity going on. So it kind of steals from both of them. So we'll put 2 on there so you know what's happening with those electrons. So this would be water, and we all are kind of familiar with that. The fact that water is polar means that, you know, polar likes to hang with polar. Then non-polar doesn't want to mix with that. And so that's where we're getting into those terms of hydrophobic and hydrophilic, water loving and water hating, we're fearing. Okay, so that's what those terms are really referring to, and that's what polarity is. And if you have a molecule that has both things going on, like our phospholipid, so if you have your PO4 and you've got all these negative charges to it, and it's hanging out as the head portion of your phospholipids, We'll just draw it like this. And all of this is a hydrocarbon tail. And that will equal hydrophobic portion. Because why? Because it's nonpolar. And then this part up here, which has clearly got a charge going on with it, this the head, the charged head, is hydrophilic. Water loving because it is charged. It's literally charged. So this one isn't just polar, it's charged. So it's fully committed. So that whether something is charged is basically essentially like saying whether it has the ability to ionize. So if it is able to literally have donated or stolen electrons, and not just sharing them unequally. So that actual charge is going to exist, and this negative charge that will be attached to this because it'll steal an electron from an atom that it would be attached to. So it's very much charged, and that these aspects on here, the fact that it has both things going on, means that it's amphipathic. Anytime you hear path, it's usually just talking about the condition of something, right? So amphipathic means we have two states going on at the same time. All right, so we also talked about pH. It's the negative logarithm of the concentration of hydrogen ions. That's what that equation means. And we know that the pH scale goes from 0 to 14 with 7 in the middle. We know that water is neutral because water can cycle between this just pure H2O or the hydrogen ions plus the OHs. If it does the hydrogen ions and the OHs, these two guys are equal. And that means it would fall in the middle. That's why it's 7. Everything that's higher than 7 is a base. Everything that's lower than 7 is an acid. And that's because what's going on in the acid end is you might have 10 to the negative 2 as your hydrogen ion concentration. Whereas up here at the base, which means you have more hydrogen ions. And down here at the base, you'll have less hydrogen ions than that 10. We're always comparing it to the 7. Okay, wow, we're really just click on everything today. It's always compared to the 7. So whether it's more or less than the 7 is where you're going to call it an acid or a base. And that's if I'm comparing it to the 7. If I were to ask you to compare a pH of 2 and a pH of 5, which one is more acidic, you should know that the pH of 2 is more acidic. And that might make sense to you. If I were to ask you from a pH of 2 and a pH of 5, which one has the most hydrogen ions? The one that has the most hydrogen ions between 2 and 5 would be 2. So you have to be able to think about it, not just that 2 is more acid, but the fact that if it's more acid, that also means it has more hydrogen ions. You must be able to think about it that way, simply just because, if anything, because I'm going to ask you that question on the test. All right. So there's that. So let me erase some of this so I have a little bit more space to write. Wish this was bigger. Little tiny eraser. I'm too lazy to click on the thing and make it bigger. Okay, here we go. All right, so for our biological macromolecules, we had 4 that we had to know about. We had our carbohydrates, we had our lipids, we had our proteins, and we had our nucleic acids. Our carbohydrates, the subunits that make up a carbohydrate are known as saccharides. A saccharide is an individual sugar. Okay. What were our subunits that make up our lipids? I don't need you to worry about it, okay? But the thing that you need to know about lipids is that these guys are hydrophobic because they have long, non-polar hydrocarbon tail chains involved. All right, so our proteins, their subunits are amino acids. We'll talk about the structure levels here in just a moment. And then our nucleic acids, this is DNA, RNA, and we also talked about ATP, our energy molecule. These that's what they are, right? Sure, those are examples of nucleic acids, but our subunits that make them up, which I skipped over completely when I was talking about it, the subunits that make them up are called nucleotides. And a nucleotide consists of a 5-carbon sugar, usually based off of ribose, which is a 5-carbon sugar. And like I told you in class, y'all getting so confused about this. And I don't know why so many of you think there's only 2 carbons in deoxyribose. I don't know where you're getting that from, but whoever taught you that taught you wrong. So deoxyribose is literally telling you what they're doing to ribose to make the difference. Okay. So ribose, which I'm telling you, is a 5-carbon sugar. Okay. Ribose is a 5-carbon sugar. Deoxyribose, all you have done is deoxied it. And what does oxy mean? It's literally talking about an oxygen. How many carbons are in oxygen? Please don't make me laugh. There's no carbons in oxygen. We're literally talking about oxygen. Okay. So we have removed oxygen. As it turns out, that oxygen comes with an H. It's an OH group that we're moving off completely. So it's a hydroxyl group that comes off, but it's no carbons involved in that. It's literally just an oxygen we're taking off. So that's all deoxy is taking out the oxygen and the hydrogen that comes with it. There's no carbon we're losing. So still 5 carbons, okay? So a 5-carbon sugar that is attached to a phosphate group. We'll actually just write out the chemical part of it. PO4, that's phosphate. And then that sugar is also attached to a nitrogenous base, which I'll put NB. And that literally just means it's a carbon, hydrogen, oxygen compound that has nitrogens in it. That's all it's telling you. Okay. So organic molecule that has nitrogens in it. And these things, there's 4 different ones of these. There is adenine, thymine, cytosine, and guanine for DNA. For RNA, you replace the T with a U. And we're going to focus on that more in chapter 6. But there's 4 different bases. The A's bind with the T's and the C's bind with the G's. And the way that I remember that is that we've got these nice, pretty little round guys here. The round goes with the round and the harsh goes with the harsh. The sharp lines go with the sharp lines. And that's how those guys are going to get attracted to each other. And they're attracted to each other. Again, C binds with G on the other side of your double helix because of hydrogen bonds. Right. Anyways, so that's how your nucleic acids are set up. ATP is our energy molecule, meaning it stands for adenosine triphosphate, is really important energy storage molecule. We'll get into how it's made and everything like that in chapter 10. But those 3 phosphates are constantly with those negative charges pushing away from one another. They're held together to one another by covalent bonds. But so whenever you break those bonds, we'll release a ton of energy, and the cell can use that to power its processes. All right, the last thing that we had to talk about were our the last thing that we had to talk about was our protein structure levels. Let's get rid of this. Make a little space for the protein structure levels. So we talked about this in terms of like drawing a ribbon, basically. So we have our 4 levels: primary, secondary, tertiary, and quaternary. Our primary is just our amino acid sequence. And I think of this as like basically writing out the word of the ribbon, right? So this is the ribbon. So you're creating the actual ribbon, spelling it out with your amino acids. Secondary structure. Now our amino acids that are next to each other, immediately next to each other, because of the differences in their R group, which the R group is what determines which amino acid is which, they're interacting immediately next to each other, creating these larger structures that could be random completely. But if you are making a larger structure, it would either be an alpha helix, which is literally just going to look like a helix, like a corkscrew, or a beta pleated sheet. And that's going to have lots of straightaways, folding back on each other, snaking along like this, okay? So those are the two major structures we can see with the secondary level of structure. If you see any of these, you should just be thinking, if you see alpha helix, you should automatically be thinking that's the secondary structure of protein. Same thing with beta pleated sheet. How are those structures made? Because of the R groups that are immediately adjacent to one another, okay? All right, so now we can take the overall polypeptide that we've made from linking our amino acids together in our long ribbon. We've curled our ribbon at our secondary structure. Now we're going to actually tie our bow. So this is usually interaction of R groups far across the other side of the polypeptide, okay? So we're having large groups across the whole structure of that polypeptide affecting one another. This could be this could be causing hydrogen bonds, charge attractions. It could be because we want to hide our hydrophobic parts on the inside away from our watery parts of the cell, even things like that. All that's going to motivate how that bow is being tied. And then our quaternary structure is when multiple bows come together to create the functional bow, okay? The functional decorational. So in that way, we could talk about this scientifically, multiple polypeptides to make a functional protein. All right, so that's the idea. So we've gone over the bigger concepts that were involved in the chapters that we learned. So now let's go over our most commonly missed topics. For chapter 1, first we have germ theory. Germ theory, again, the theory that Louis Pasteur came up with to disprove spontaneous generation. He said there's something invisible that's causing this stuff to happen. I'm going to call them germs. That's germ theory. So miasma, just a bad smell, or things just coming out of nothing, weren't the cause of these things. There were actual microscopic organisms that we just couldn't see that were causing this. And they might need oxygen to live, and a lot of them do. So he provided them oxygen, but he made it so that they couldn't actually get in there. So if he could boil away and sterilize that broth and keep new microbes from getting back in, it's not oxygen that's causing the problem. It's not the fact that there just is air. No, there has to be microbes or organisms that are getting in there. And that's Louis Pasteur that came up with that. So obvious to us now, but he's the one that came up with that guy. Yes, you're going to have a question on the exam about him, okay? So that's why it's the most commonly missed one of the ones, because it's actually on the exam in the past. All right, so Pasteur is the one that came up with that. Pasteurization, like we do to our milk, it gets rid of germs. So if you can remember that pasteurization gets rid of germs, maybe it'll remind you, Pasteur, who pasteurization is named after, is the one who came up with germ theory. Here's an example. Who developed and tested germ theory? You would have to know that it was Lister, or excuse me, that was getting Lister in just a minute, believe me. Pasteur that did that. Pasteurization, germ, getting rid of germs, germ theory. Okay. So be able to pick their names out of a lineup. You do have to know who Lister is. We talked about Lister and how he's the guy that used, chemicals to sterilize his operating theaters. So he actually used sanitization for the first time. And that concept of preventing that contamination, he's kind of the father of applying aseptic technique. You don't need to worry about Edward Jenner right now. We'll talk about him later. Edward Jenner came up with the first vaccine, just FYI, the very, very first vaccine ever, which was for smallpox. We do not vaccinate for smallpox anymore here in the United States. Russia does. I'm just going to leave that comment there. All right. And then Einstein is not going to ever have anything to do with microbiology, so you're not going to need to know him. You do need to know Lister and Pasteur for Unit 1. They're, in fact, the only ones that you're going to need to know for Unit 1. All right. Plague doctors stuffed the nose of their masks with herbs and flowers to ward off bad smells. They thought those bad smells were causing the plague. Literally, they thought the smells were causing it. And that's called miasma. They had a term for that. So they put flowers and herbs in those long noses. That's why they had long noses on the masks. And so they'd stuff them to prevent from smelling the smells, because they thought that would prevent them from getting sick. So why was that ineffective? because preventing the smell is not what's causing the disease, right? So already I'm assessing this question, and you should always be assessing all of your questions on your tests and quizzes before you even look at your choice. Why wouldn't herbs and flowers prevent you from getting sick? Because smells aren't what causes you to get sick, right? It's germs. So, microbes. So, let's see our choices and see what aligns with that type of thinking. This would have worked if the herbs were antibacterial. Interesting. We'll come back to it. Smells do not transmit illness. Yes, this is applying to what we're thinking about now. It's literally what our line of thinking is. It blocked them from seeing their patients clearly to treat them effectively. That's not why it didn't work. That's not why. It's not why herbs didn't work. Even if they could have seen their patients, the herbs still wouldn't have stopped it. The contaminated herbs were being transported from home to home. That's not why the doctors were getting sick. The homes already had the sick people in them. The homes already had the sick people in them. Okay. So don't get confused there. So here we have, this would have worked if the herbs were antibacterial, or the smells do not transmit illness. Even if the herbs had been antibacterial, so the plague is transmitted by a bacteria. Okay. But this isn't a good reason for why this was ineffective. It's ineffective because smells themselves aren't what transmits the illness. It's bacteria that's causing the illness. And these people were sick, and they were coughing and stuff like that and transmitting this illness in the air. We also know that fleas were usually the ones that were primarily transmitting the illness. And so they would bite you and transmit the bacteria into your literal bloodstream. That's not going to prevent you from getting sick, right? So that is why this part isn't accurate. You would know a little bit about that if we were talking about the plague itself and learning about the plague, which we will in later chapters. In chapter 20, I believe, 21 we'll be learning about that. And if I thought that was important on your test, I would have included that information. This is not a test question, though. This is just to help us start thinking about this stuff. But anyways, my whole point here is that there are also going to be diseases that aren't bacteria. And I hate to break it to you, antibiotics, for example, there's a word, so this says literally antibacterial. I hate to break to you, there's diseases that aren't caused by bacteria. So this isn't going to protect you from everything. And bad smells can come from things that are decaying from fungi and yeasts and things like that. For example, I hate to break to you, but those aren't bacteria. Okay, those are different organisms. Viruses also can transmit disease. And even if a person was sick, let's say with the flu, and that person's been throwing up and they smell like vomit and they smell bad because they ain't been showering and all that. Even if you hit all those bad smells with good smells, can you still get the flu from them? Of course. So that's like part of what I want you to take home from that. Smells don't transmit the illness. The microorganism does. The pathogen does. So our next one was talking about eukaryotes versus eukaryotes. A being without, karyote being nucleus. Okay, so that's what's going on here on this slide. And then U means true. So the literal definition is in the name, if you can remember what those parts stand for. So A, like asymptomatic, without symptoms. Eukaryote, without, karyote meaning nucleus. U means true. So, try to think of a good example of that, but I don't know. I'm not coming up with one just off the top of my head. But anyways, also you can think of A comes before E in the alphabet. It's your little memory device down here. A comes before E in the alphabet. So in evolution, eukaryotes would have come before the eukaryotes. And that means they're probably simpler cells. So they're not going to have as many structures, including the nucleus. All right. It's more primitive, more basic. We can break it down that way to help us remember. So it is believed mitochondria evolved from a bacterial symbiont. So what that means is that there was a cell that ate another bacterial cell, and they ended up being able to work together so well that over time, that cell that it ate up became a mitochondria. There's a lot of evidence that supports that that is what happened, including the fact that mitochondria have their own DNA. You ever heard of mitochondrial DNA? You probably have. Your mitochondria have their own genome, and your genes in your actual genome inside of your nucleus that contains all of your genetic information, it doesn't count. It does not code for your mitochondria. Your mitochondria code for themselves. And it's the only structure in your entire cell that does that. And it has a circular genome like bacteria do. We have X's, they're linear. It's linear. It's a line. It's not a circle. So we'll get into all other reasons as well in chapter 4 and 5, but it's believed that mitochondria, our powerhouse organelle in our cells, actually they were originally bacterial cells that were just ingested by another cell, and they found out a way to work together. That's called symbiosis. If you can't have eukaryotic cells without mitochondria, what can you infer? So eukaryotic cells get their energy from their mitochondria. They don't make it any other way. So you can't have a eukaryotic cell without their mitochondria. So what would you infer from this idea of how this happened? Did bacteria evolve from eukaryotes? Did eukaryotes exist before bacteria? That wouldn't make sense. Clearly, this setup is telling you, now that we're starting to look at our choices, that if eukaryotes came from bacteria, which one came first? Bacteria first, right? So bacteria existed before eukaryotes is our third choice, or eukaryotes evolved from archaea. This isn't insinuating that. From this information that we're getting here, the best answer for this question will be C. Bacteria existed before eukaryotes. Next question. Looking in a microscope, you see a large cell with several membrane-bound sections, little compartments that are surrounded by membranes. What type of cell would this potentially be? Could it be yeast, archaea, bacteria, viruses? In chapter 1, we learned that a virus is acellular. That means without a cell. Literally, we already talked about what A means, without. So it can't be that. What type of cell? Can't be that. All right. So if you're seeing membrane-bound organelles, that would include a nucleus. If you have any membrane-bound organelle, you also have a nucleus, bottom line. That's just the rules now, okay? So do archaea or bacteria fall into that category? These guys are both eukaryotes. So without, and then karyote being nucleus. So it's literally telling you it doesn't have a nucleus. If it doesn't have a nucleus, it doesn't have any other membrane-bound organelles. So it's not these guys. So even by process of elimination, you should get to yeast. But we'll actually be talking enough about that in chapter 5 that you will just know that yeast are fungi. Fungi are eukaryotes, okay? We only have 3 domains: eukarya, bacteria, and archaea. That even tells you that bacteria and archaea are not eukaryotes. Okay, eukaryotes are the ones with the membrane-bound stuff. All right, so for chapter 3, one of our most missed topics was the difference between selective and differential media. Again, selective, selecting for organisms. Certain organisms will grow on it, others won't, has something in it to prohibit the growth of other organisms. Differential media is going to let you see the difference. It's always going to be based on metabolic or biochemical abilities of those organisms. And so the byproducts are usually what that media color change is detecting. So differential, yes, you're going to have color change. But what it's really telling you about is the biochemical or metabolic difference. Okay, to tell you, but your metabolism is just another word for all of the biochemical processes that are going on in a cell. They're the same thing. Okay, so we're just talking about biochemical differences, and it's usually some sort of like waste or byproduct that we're detecting. Okay, selective, again, this guy is just going to allow some to grow and prohibit others. And vice versa. You can have media that can prohibit groups from growing, and then also assess at the same time a color change of those that did grow. So you can get a lot of information from those types of media. Okay, so I'm going to move on. Oh, here's our our memorization thing. Selective selects the ones to grow. Differential tells you the differences, but we're not selecting things from growing or not. All right, our example questions. Which of the following is an appropriate definition of selective media? Is it, so we know selective media is media, before we get into our choices, selective media, some will grow, some can grow, others are prohibited. So now we can start looking at our choices. Media that changes colors due to metabolic byproducts, you should be looking at that choice and being like, nah, man, that is differential. Media does that. So it's not this one. Media that favors the growth of some organisms inhibits the growth of others. That's literally our definition. Media that selects for organisms which require added nutrients. So those added nutrients that they would require, do you guys remember the term we use for that? Growth factors. And media that has growth factors that might be required for some organisms is called enriched. And those organisms that need that, they're called fastidious. Right? So it's not this one. And then we have media that kills all bacteria except for one type. We're not talking about killing bacteria. We're not talking about just one type of it. So the more general approach, this is the more correct answer. This is getting into a weirdly specific category that not all of our situations would apply to when we're talking about selective media. So B is our most correct answer here. All right, so McConkey agar, it contains bile salts. This is my example of what you will see on the test, okay? This is what I mean when we start getting to those critical thinking questions. This is a great example. So I'm going to give you the information you need. I don't expect you to know what McConkey agar is. I'm going to tell you about it. Now you're going to answer some questions. McConkey agar contains bile salts that favor gram-negative bacteria and inhibit gram-positives. Okay, so we know we're favoring something and inhibiting something else. That's selective media. Simultaneously, it has an additive that causes bacterial growth to turn pink if it can break down lactose. Well, that part of it, we're not inhibiting or prohibiting with that, but we are having a color change if we can do something metabolically or biochemically. So this is differential. So what type of media is it? Is it selective? It is, yes. Is it differential? It is, yes. So the right answer would be C. It's both of those things. Okay. Next question, or next topic, chapter 3, growth factors. We've talked about it already a little bit. A growth factor is required. This is something that the organism can't make. And so it needs it in its food. And in this case, our food is its media. Okay, that's what our microbes grow in. So enriched media is the media that has the growth factor in it. And bacteria that need growth factors are called fastidious. So remember those three things. The growth factor, it's like our vitamins. All right, the media is called enriched media. Has it in it. And then fastidious is the actual organism that needs the growth factor. Okay, so that's how we'll break that down. One way you can try to remember that growth is a factor you can't live without. That kind of gets you thinking about the fact that the growth factor is something that you can't live without being given to you, okay? All right, so which scenario details features of a fastidious organism? So what does fastidious mean? Fastidious means it requires a growth factor. So if you're always going to be thinking about fastidious, you should always be thinking about all three terms. Fastidious organisms will require growth factors in their enriched media. I haven't even looked at my choices yet. So you should be thinking about your questions like that before you look at your choices every time. So we have that written down as our note for this question. So now let's look at our choices. Bacteria that turn pink when they grow on McConkey agar, cows, which are herbivores, velociraptors, which require lysine in their diets. So that would make that a growth factor, wouldn't it? They have to have it added into their diets. They would be living, just Jurassic Park. And then archaea, which grow in extreme environments. Only one of these scenarios talks about the organism requiring something being added into their diet, and that is C. So that's the only correct answer here. The other ones don't talk about a specific nutrient required to be added to their diet that they can't make for themselves, okay? All right, next one. Which of the following could be considered enriched media? All right, so we're looking for media. If it's enriched, and again, we could look over here. We already made one note about it. If it's enriched, it has a growth factor in it, right? So let's look for one that talks about that. McConkey agar, bile salts that inhibit. This is not talking about a growth factor being added. Simmons citrate agar. Only certain kinds of bacteria can utilize citrate as their sole carbon source. Now, this is a great biochemical test. This would be a type of media that we would be considering to be selective as well. But it's not talking about a growth factor added to it. Nutrient agar, a basic medium on which a majority of bacteria can grow. There's no growth factor involved in that. Anything can grow on it, but there's no specific growth factor added. And then blood agar. It contains nutrients that some bacteria need and can't get from other media. That's literally basically our definition of what a growth factor would be. That's our enriched media. All right, moving on. We only got a couple more after this and then we're done. Okay, I promise. All right, our next topic, the differential stains. Staining protocols that allow you to tell bacteria apart based on whether or not they have certain structures. So these are stains. Differential stains are tell you whether your organism has a structure or not. We'll say a feature. That's even better. By looking at it in a microscope, okay? So does it have a capsule? Is it acid fast? And we'll talk about what capsules are and acid fast. That's coming up in chapter 4. You don't need to have these specific tests down pat how they work right now in chapter 3. In chapter 4, you will though, okay? And when we do it in the lab, you're going to need to know these stains, inside and out. But for now, just understanding that this is the concept of what a differential stain is. Gram stains tell you, literally, you'll do a gram stain, and if it is positive according to the rules of the gram stain, you are a gram positive organism. If it is negative according to the rules of the gram stain, you are a gram negative organism. Turns out there's certain traits that are contributing to whether you're positive or negative. And we'll talk about those in chapter 4. All right, so some stains will work based on charge of the stain itself. And so we've talked about the positive stains being attracted to the negative cell surface and the negative stains being repelled by the negative cell surface, so they'll stay in the background. So that's down here as well. It's commonly missed on my exams that negative stains will be staining the background. All right, and that positive stains will be staining the cells. And this is just explaining that. So here's a little memory device. Let's see. Negative stain and negative cell both have negative attitudes, so they're not going to get along. So that's how you know they're going to repel each other. Let's get into some questions. Which of the following would be considered a differential stain? Staining for the presence of flagella. Does your organism have a flagella or not? This is pretty much how I defined it, right? It's tell you if your organism has a feature or not. I literally wrote that out over here. So differential stain tells us if we have feature or not. So if flagella, which is something that bacteria use to get around, that's the feature. So this is probably our best candidate. Let's go through our other choices. Stain using one dye that stains one color to create contrast. These would be our simple stains. It does create contrast, but does it tell you if you have a feature or not? It doesn't itself, right? A stain which kills bacteria first doesn't tell you anything about the features, whether it has a feature or not. And stain a slide with one dye and then have another slide of the same organism, stain it with a different color dye, and then compare them. That is not a single stain that's going to tell you a difference in that one stain. The whole point of these differential stains, you're doing it on that one slide. You put it through that whole experience, and based on its final color and final colors that you see in that slide, on that one slide, all treated that one way in that staining procedure, that's a differential stain. It's all happening on that one slide. So that's going to be staining for the presence of flagella. Does it have flagella or not? That's going to be A. All right, our next question, mycolic acid. We're going to talk about this more in chapter 4, but mycolic acid, I'll tell you about it, is a wax on the surface of acid-fast bacteria. Why might you guess that a special differential stain, carbofuscin, would need to be used to stain positive cells for acid-fast stain? This is something we will definitely talk about in the lab more on how this works, but now we're getting to think about it ahead of time. So if mycolic acid is a wax, and we learned in chapter 2 that that wax is going to be hydrophobic, it actually falls into that lipid category, right? We had it listed within our lipid group of our macromolecules. So carbofuscin is help us stain mycolic acid. So we know mycolic acid is going to be stained by carbofuscin. That's what it's telling us. Acid-fast bacteria have mycolic acid. We stain it using carbofuscin. So carbofuscin, is it a positive stain? Is it lipid soluble? Is it heat activated? Or is it a negative stain? If we're staining lipids, positive stains have a charge, y'all. Does a lipid care about a charge? It hates it. It's not going to stick to that. Is that lipid going to be stained by a negative stain? No, it hates it. It hates charges. It's repelled by that. Nonpolar versus polar? Are you kidding me? Forget it. So that's out. Those are out automatically. Lipids that are hydrophobic, they are nonpolar, are not going to associate with polar molecules. They're going to be repelled just like your oil is repelled by your water. So we can knock those out right away. So probably, I mean, obviously, I feel like this speaks for itself, right? If we're talking about a lipid, how are you going to stain a lipid? You want a stain that's lipid soluble. So you will be able to pick that choice based on that, okay? All right, 2 more, literally. We have gram stains. We're going to talk more detail about the gram stain in chapter 4. So this probably doesn't really necessarily belong here, but it is commonly missed. So gram stain is a differential stain. And in the gram stain, purple means positive, P positive, purple. Red is a negative, what I think is a negative color. That's also what the negative color is, this, okay? So purple would be positive, red is negative, and that's because purple is imparted by crystal violet. And it sticks in the walls of our gram-positive organism. They have thick cell walls. So our thick wall gram-positive versus our thin wall. Sorry, gram-negative. That crystal violet sticks in here. We'll talk about this in chapter 4, but it used to be I talked about it in chapter 3, so that's why this is popping up. I forgot that I don't talk about it in chapter 3 anymore. But anyways, to preface you guys for it, so you'll understand about it when we get a little better, that red color is imparted by saffron, which is our counter stain. because we're going to rinse out the crystal violet of our gram negatives when we rinse it with alcohol. Anyways, I don't want to get too much into the steps of it because we're going to learn about that in chapter 4. But the idea is the crystal violet is our differential stain. Gram positives are purple. Gram negatives are red. Red's a negative color, and that's how you'll remember that. So what is the difference in gram positive and gram negative bacteria that makes them stain the way they do? And I just touched on it a little bit. That the gram positive, because it has that thick wall, traps the purple in it. Okay, just literally just traps the purple in it. So why does it trap the purple? It's because of the charges on the cells, the presence of LPS, which is lipopolysaccharide. We'll talk about this again. This is a chapter 4 thing. The presence or absence of the nucleus, or the difference in that bacterial cell envelope. So the cell wall is part of the envelope. And so this is going to be our answer here. And our patient sample is sent to you. And like I said, the test isn't going to tell you that it's a chapter 3 question. And when you get to unit 1, you have to know chapter 4 and 3. So that's how these chapters are going to relate to each other. So I apologize that we haven't got chapter 4. This definitely applies more to chapter 4. But a patient sample is sent to you testing for the presence of a suspected gram-negative bacteria. After performing this gram stain, your cells are purple. We already looked at this. Look at this. It's purple. It's positive. So, what are you going to tell the medical team? They thought it was going to be a gram negative, but it turned out it was purple. Are you going to tell them that the sample is in fact gram negative and they're correct? Are you going to tell them the sample is gram positive, so they were incorrect? Yeah, that's what you're going to tell them. But what's our other choices? They must have sent the wrong sample. Please. You're not going to infer that. If you are doing your job and you're doing it the way that you're supposed to, you shouldn't just be assuming, oh, they must have sent the wrong sample, or, oh, this must be inconclusive. They thought it was going to be gram negative and I got gram positive. No, you're going to tell them this tested as gram positive. This is more or less thinking along the lines of being sure that you're doing your job correctly. And if you're doing it correctly and this is the results you got, sometimes the medical team didn't necessarily guess the right organism. Maybe it's more likely it would have been gram negative, but this test is showing it's gram positive. That's going to help them treat their patient better. So this is coming from the lab side, and we're learning about that lab side. You might be the other person on the other side of that in the future saying, I think my patient has a gram positive disease, and the lab is telling you it's gram negative. You don't call the lab and tell them they did their job wrong. They're telling you that it was gram negative, then you would run with that. So this is important to think about. If that's being done correctly, that everything's done right. All right, chapter 2, list concept with our chemical bonds, being aware of the difference between covalent, ionic, and hydrogen bonds. Remember, covalent, we are sharing electrons. That's pretty much all I have written here. The atoms are literally sharing. It might not be equal sharing, but they're having to share electrons. So covalent, we're sharing. In ionic, we are transferring or donating electrons. And hydrogen bonds, these are all attraction based. And it's always going to involve hydrogen at some point. Okay. So literally a hydrogen being attracted to another thing. And the hydrogens are never, ever going to have a negative charge. They're all, or polarity. They're always only ever going to have a positive polarity being attracted to something that is negative polarity. So that's what's going to be happening there. So here we have down here, people share COVID just like electrons are shared in covalent bonds. Okay. COVID, covalent, sharing. Okay. Hydrogen bonds are when hydrogens just say hi to other atoms. I like this one when I wrote that one. I thought that was a cute one. Hydrogen bonds are when hydrogens say hi to other atoms. And opposites attract, so they're saying hi to the negative polarity or charges. So let's go to the question for this one. The bond between magnesium and sulfate in magnesium sulfate allows it to dissociate into Mg plus and SO4 minus when dissolved in water. What kind of bond exists between Mg and SO4? So these guys dissociating into their little charged things, we only see that happening whenever we're able to completely donate permanently an electron to another molecule. And this situation when we have them dissolving and separating within water because of that nice, happy polar, we all have charges and polarities party together, right? So the polar party. This is only happening because of ionization. And so we're looking for ionic bonds here. Okay. All right, the next question. DNA exists as a double helix, and it's called double because there's two strands held together by hydrogen bonds. Okay. In any one of those given bonds, what type of electron interaction is occurring? In a hydrogen bond, what kind of electron interaction is occurring? Are electrons being shared? They are not. Are they being donated in a hydrogen bond? No. Are there polar attractions with no electron transfers? This is starting to sound like my choice. Or are there negative electrons becoming positive? Now, this is ridiculous. Your electrons are not becoming positive ever. Forget it. Electrons are negative, period. So it's not this sharing of electrons. That would be a covalent. It's not donating of electrons. That is literally the definition of an ionic bond. So we are going with polar attractions with no electron transfers. And that is it for our review for week 1. It went a little bit over, and that's okay. I'll try to keep the other ones a little bit shorter. Thank you guys for coming. If you're still here, let's see who we've still got on here. Oh, still got a few people here. So thank you guys for coming. If you're still here, that's it. If you are a Monday, Wednesday person, I will see you on Wednesday, probably, right? The reason I'm saying bringing that up is because of weather is supposed to roll in on Wednesday. So I'll probably see you on Wednesday. I'm more concerned about my Tuesday, Thursday people. Remember, Tuesday, Thursday people, we do not have class on Tuesday to keep you on the same schedule as my Monday people who aren't having class because of Martin Luther King Day. You would otherwise have Tuesday, Thursday people, you would otherwise have an extra class day compared to our Monday, Wednesday people. So we're keeping that even. We have Tuesday off for my class. I will still be in my office on Tuesday during when I would normally be on campus, except for my night class. I won't be showing up during that time. However, on Thursday, when you might have, class, lecture and lab for chapter 4, lecture, and then lab would be our lab safety lab. If we have a weather issue, please keep a lookout for an announcement on what to do if the campus closes. So OSU will send you notifications on the closure, and I will send you an announcement that will let you guys know about what you need to do on your end. So that is it. I'm going to let you guys go. And I believe, let's see, yep, no problem. I'm glad to help you guys and stay. So Maria, I believe that you wanted to have a discussion. I'm going to send you an email with a different Zoom link so that nobody can sit in on our conversation. So I'll send you that right now, just as soon as I log off of here, and I will post this recording in the discussion session as soon as I can get it up on YouTube. So definitely within 24 hours. And email me if you guys have any questions, and have a good weekend. Be sure you get your stuff turned in by midnight tonight. Anybody have any questions? I'll let you go.